Hello students, welcome to the next lecture of our third week SWAM course. In this we will start about our discussion with complex permanent tissue. We have previously discussed about simple permanent tissue which were made up of only one cell type. Now we will move on to complex permanent tissue which are made up of different cell types. And in this the two important complex tissue are xylem and phloem, and they are basically the conducting tissue which are found in the vascular system. So, first of all we will start with xylem. Now, see when the plants were evolving, okay, earlier plants were found in the water because and they had a very simple body organization, and when the plant evolved to land plant, the most important requirement was to develop a proper vascular system. Only then they will be adapted to the land systems. So, a development of a proper vasculature was a major necessary which led to the advancement for the colonization of land plants, and land plants have two primary conducting tissue because on the lands you require that you take up water from the soil and then you conduct to different parts of the plant because every part of the plant is not exposed to water. So, they require water for their survival, and for that proper conduction is required. Now, in plants two primary conducting tissue are xylem and phloem. Now, xylem, as you all might be knowing, that it is responsible for the transport of water as well as the dissolved minerals which are present in the soil. So, along with the water, the minerals also get transported to different parts of the plant. Phloem also, it translocates the product of photosynthesis. So, basically xylem is conducting water and it is transporting to different parts of the plant. It is giving to leaves where photosynthesis is occurring, and then photosynthesis is preparing food for the plant. Okay, it is converting light energy into chemical energy. Different forms of carbohydrates and different forms of other biomolecules are formed, and that is all then translocated or moved on to different parts of the plant because every part of the plant will require food for their survival. They are performing lot of metabolic activity, so they require food, but yes, they are not all manufacturing. So, manufacturing of food is done by leaves as well as green stems. So, from there this food is translocated with the help of phloem. Now, they are complex tissue because made up of multiple cell types, more than one cell type, and so their role is definitely in transportation, in communication also, like along with transporting the various nutrients which are required for making the food for their survival. Some other communication and coordination is also required between different parts of the plant. That could be done with the help of like phytohormones, and many other molecules are also there. So, they will also move through this conducting tissues, and they will be communicating among themselves. So, communication and coordination, and they also provide support because they are basically made up of, many are made up of dead tissue. Hard tissues, highly lignified tissues, so they will also provide mechanical support to the growing tissue. See, when the plant is growing, when a big tree is formed, a lot of secondary growth occurs, the diameter increases. So, in that basically xylem and phloem tissues are increased, and many of the tissues are not only to increase the conduction to the growing tissue, but also to provide mechanical support to the growing and increasing part of the plant. Now, coming to xylem. Now, the xylem term was given by Nageli, and it is derived from the Greek word xylos, which means wood. So, xylem, it is a complex tissue which its principal role is to conduct water and also to provide support. It is more sclerified, that means it is more lignified, more secondary wall layer deposition as compared to phloem. Now, developmentally two types of xylem is there, primary xylem and secondary xylem. See, when we were talking about different types of tissues in the earlier classes. We have discussed that there are primary tissues which are present from the growth of the plants itself, from the time when the plant is growing from the embryo. From there itself they have primary tissues and they are present till the end of the plant and they are derived. So here primary xylem will be there. So primary xylem will be derived from procambium, from the apical meristem, which give rise to the primary tissues which is present in shoot apical meristem and root apical meristem, and there also you have a procambium which give rise to vascular tissue, then you have protoderm which give rise to epidermis, and then you have ground tissue which give rise to cortex and pith. So in that, procambium will give rise to primary xylem and it will be developed during primary growth of the plant. Then secondary xylem will also be formed, but it will be formed later in the development of the plant from cambium. This cambium was produced from procambium and during the lateral growth of the plant. Now in the primary xylem, the primary tissues, you have two types of xylem, protoxylem and metaxylem. See, protoxylem and metaxylem, they are all primary tissues, means they are formed from the early developmental stages, but in this you categorize protoxylem which is the first form xylem and which is formed when the plant is still growing and it is found in young leaf and stem because these are still growing and they are in the division phase. So that time the xylem which is formed is known as protoxylem. In this the cells are small, okay, the cells are small and the secondary cell wall is also very less, and in this you will find annular or helical thickening. See, when we were discussing about deposition of secondary cell wall, we have discussed that this cell wall will be deposited in various patterns, like helical pattern, like this is the primary cell wall in which in all around the cell in helical fashion this secondary cell wall will be deposited, or in this annular fashion then you will have helical fashion, okay. So at certain locations, so in this all the points of the primary cell wall will not have secondary cell wall. Then you will have like pitted fashion also, in which there will be a continuous deposition of secondary cell wall forming pits at certain points, means pits are the only holes where secondary cell wall is not deposited, otherwise it is found on all the cell. Similarly, you will have reticulate, okay, net-like and scleriform, ladder-like. So this all we have actually discussed before. So net-like or reticulate pattern, then you have scleriform, then ladder-like pattern. So protoxylem will have annular or helical thickening because in this case the cell wall is not so lignified in this pattern, and this will leave a margin of still some growth. When the xylem tissue is still growing in the early stages, then this type of thickening is there, so that the other portions are still not so hard and they have a chance for increasing. Then in this protoxylem you will find the tracheids and phloem vessels you will find, in parenchyma you will find, however it will not contain the fibers that we will see later. Then metaxylem is the later form xylem. It is found in mature plants when the plant has somewhat attained its development and it is not so elongating. So this will also contain fibers. This metaxylem will contain fibers. These are more thick walled cells because it has less chance of elongation. More thickening in the form of scleriform and reticulate thickening or pitted thickening will be observed. Then coming to elements of xylem. Now 4 different types of xylem is present, which is tracheid, vessels, xylem fiber and xylem parenchyma. In this tracheids, vessel and xylem fiber, they are dead tissues, and xylem parenchyma is the only living tissue which is found in xylem. And in this tracheids and vessels have the conducting role, whereas xylem fibers, they are just for support. See, we have discussed about sclerenchyma, the fibers. So this is the xylem fiber which is sclerenchymatous and this is required for support. Coming to tracheids. Now, tracheids are, it is found in primary and secondary xylem. That means in the early stages in protoxylem and metaxylem, primary means it is protoxylem as well as metaxylem. Okay. This is found in primary tissue. Tracheids are also present when secondary xylem formation takes place from Cambium. So, in secondary tissues you will also find tracheids. Now, they are hard, thick walled with pitted wall. Now, tracheids are also, you can say that they are also long with tapering ends. Now, when you draw tracheids, they are long, okay, they are long and they have tapering ends, means their end wall is covered, okay, there is no opening in the end wall, and when you will observe them under cross section, you will observe them that they are angular in appearance with thick walls, okay, thick walls are there, and they are single cell. This is a single cell, okay, this entire tracheid which is shown here is a single cell, and they will be parallel to the vertical axis. Like suppose this is the stem and this is the vertical axis. So, these tracheids will be parallel to the vertical axis and they will overlap with each other. Now, this tracheid, now lot of tracheids will be overlapping with each other. Okay. So, they will overlap and they will be very adjacent to each other, and this overlapping will help them in communication. Now, they will have pits, okay, this pits will actually help in communication, but since they are lying very close together, they can easily move. Now, these pits will also be present on every side of the wall, every side of this xylem, and these pits may be simple, like simple pits, okay, or they may be bordered pits like that, that all we have discussed, and half bordered also we have discussed, that when a pit of a tracheid lies with a parenchyma, so on parenchyma cell actually no pit will be there, whereas on the xylem side pit will be there, so that pit will be known as half bordered. So, bordered pit also we have discussed. Lignin will not be deposited uniformly, as a result different types of pit sculpturing will be there. So, that also we have studied, like annular and spiral thickening will be there, which will be observed in protoxylem, and then scleriform or reticulate pitting form will be there, which will be found in metaxylem. Scleriform pitting, actually this is not the deposition. Pitting, when we talk about scleriform pitting, means ladder like pits will be observed, okay. Occurs in all vascular plant. This tracheid is found in lower plant groups also, like in pteridophytes also, in gymnosperm also, and in higher angiosperm. So, this tracheid is found in every plant type. Now, how actually this pit will move? See, this pit have connections, okay. Suppose we are drawing pits here. This is having connection with other pit, with other tracheids. Now, pits, suppose water is flowing, conducting, see this is also a conducting tissue, so it will conduct water. Suppose water has entered in this tracheid through the pits, they can move in another tracheid with the help of pit pairs which are attached with each other. So, in this way they can move and they can cross the tracheid. So, in this way they will actually flow in the upward direction by with the help of pit, the water can move from one tracheid cell to another tracheid cell. Now, coming to vessels. Now, vessels are more advanced form of tracheary element, means conducting element present in the xylem. They are basically found in angiosperm. They are not found in lower groups of vascular plants like pteridophytes or gymnosperm, but yes, some exceptions are still there, like in few advanced gymnosperm like ephedra, welwitschia, then netum, these vessels are present whereas in some pteridophytes also, like in Selaginella, Equisetum and few more genera also, this vessels are present. Now angiosperm, there are some angiospermic families also in which vessel is absent, like in Vinteraceae, Tetracentraceae, Trochodendroceae, and in some more families these vessels are absent. Now vessels will also be present in primary and secondary xylem, and they will also originate in the primary xylem, they will originate from procambium, and in secondary xylem they will originate from cambium. Now vessels are also long, they are actually long tube-like pipes. Okay, you can see here that these vessels are long tube-like pipes, okay, and they are conducting elements. They have thickened walls, they are non-living, and the thickening, they will also have patterns like annular patterns will be there, then you have angular patterns, then scleriform pattern, reticulate. So they will also be present in protoxylem and metaxylem. So their main purpose is conduction and mechanical support. Now each vessel element is composed of single unit cells which are known as vessel members or vessel elements. Okay, this is vessel members. Okay, like tracheates was a single cell, but vessels are made by joining unit cells which are known as vessel members. So large number of these vessel elements will join together to give rise to a tube-like pipe. Now these vessels are quite broad, okay, they have very broad diameter as compared to tracheates, so their conduction is more better, so that's why they are more evolved, and it is found in angiosperm. Now each vessel element is, you can see it's quite wider, and many a times even the each vessel element can be more wider as compared to their length. So in that case they can appear as drum shape also, okay, but yes, the size and shape will vary, their length will vary. Now the important feature of this vessel element is that their end walls are perforated. See, each cell will definitely have a wall, but when they are joining in this way, their end walls get dissolved. Okay, so when you will observe them as such, you will see that their end walls are completely degraded and they are forming a continuous channel in which the water can move. So you can see perforations are there, but this perforation is very big and their end walls will form a continuous channels. Sometimes their perforations, like if they can be in an oblique manner, or they can be straight in this way, or they can be slightly oblique. Okay. Sometimes the perforation, suppose this is the end wall, okay, so perforation is completely there, a single hollow tube is coming out from here. Sometimes perforations can be sclerified, some big holes could also be there, but still it is quite wide open to allow the flow of water. Now xylem parenchyma, when we talk about xylem, it is the only living tissue, it is present in primary and secondary xylem and originating from procambium and cambium. Now xylem parenchyma is responsible for storage also, and it helps in transportation in the radial direction. Okay, so they will, two types of xylem parenchyma will be there. One will be on the radial axis, on the axial, along with the axis, they grow along with the axis, and second parenchyma will be on the radial side. This will help in radial conduction of water. Sometimes, although since they are parenchymatous, they will be thin wall made up of cellulose, but sometimes lignin deposition can occur in later stages. They actually store starch, fat and many other substances. Maple syrup is a well-known product of xylem parenchyma. It also helps definitely through radial conduction. It helps in transport and sometimes provide mechanical support through turgidity. Now coming to xylem fibers, that all we have discussed before, that fibers are elongated cells, okay, and they do not actually, they are just responsible for providing support. They are tapering cells, elongated tapering cells. They have thick lignin deposition, very reduced lumen, And these types we have already discussed. They will be libriform fiber, fiber tracheids and gelatinous fiber. So, thus we have already discussed in sclerenchyma that these type of fibers will be there in xylem. Now, this is a microscopic image where you can see broad vessels, okay, TS of a stem, which is showing you all types of cell. You can see vessel elements here, then small tracheids are there, okay. It is a cross section, okay, when you are just cutting it as such and observing it from top. So, you will see vessels are long tube like structure. So, when you cut a pipe on the top, you see a circular pattern. Similarly, tracheids are there, and these are the radial parenchyma which are present in this layer. Now, one very important role of xylem is conduction of water. Now, one very important theory is proposed to explain the conduction of water is the cohesion tension theory. Now, xylem actually is taken up from the soil, okay. In soil, lot of water molecules are there, and they will be, suppose this is the root, okay, and here lot of root hairs are there which will absorb water, water molecule is there. So, water molecule will enter the root hairs through osmosis, it will enter here and then it will enter the xylem vessels, okay. Through osmosis means movement of water from higher concentration to lower concentration, okay, through a semi-permeable membrane is known as osmosis, and then it will enter the xylem vessel, and through this xylem vessel it will enter and finally in the stem, and then it will move in the leaves. Now, what happens here actually, the transpiration, okay, transpiration is the major driving force which helps in pulling the water upward. Now, leaves are, water is evaporating from the leaves through transpiration. So, the water is actually it creates a negative pressure at the leaf surface. Actually, the water is very high in the soil. So, water potential is very high, free energy of water is very high in the soil, whereas water is low in the inner cell, in the xylem and finally in the leaves. So, this all creates a negative pressure, and because of this water enters inside the cell and finally it moves upward. Now, in the xylem what is happening? There is a property of water which is known as cohesion and adhesion. Means water molecules can stick together to their own molecules as well as they have a property to stick together to the surface. So, this sticky nature will help one water molecule to pull another water molecule when they are in a tension. Means when transpiration is going on and the water is pulled up in the upward direction and it is evaporated from the leaf, this cohesion and adhesion properties of water will help the water to move in the upward direction and through the xylem of the root and then finally to the xylem of the stem, and then finally they will come into the leaves and they will be evaporated in the outer environment. And it is said that it is the difference in the energy of the water level in the soil as well as in the environment which creates a negative pressure and because of which creates a potential difference and because of which the water moves. So, with this actually I end up with xylem. In the next class we will talk about another conducting tissue which is phloem. So, thank you for today.